Okay, great. Hello, everyone. I'm Melody Chan. I'm chairing the afternoon session. And just a reminder, if you have a question, we will get you a microphone, and please speak directly into it so that everyone on Zoom can hear clearly. Wonderful. So today we have Seth Sullivant from North Carolina State University, who will be speaking on phylogenetic algebraic geometry. Looking forward to your talk.
Okay, thanks. Well, thanks so much to the organizers for the invitation and for organizing this conference. I've always felt like there should be something like this, and I was so happy to get the invitation to come. I'm going to be talking, maybe, is it a little echoey, or there's going to be a little bit of feedback? Let's try. Okay, what should I do? Further down? Okay, can you hear me? Yeah. How closer? Take my name tag off? Is that what you said? Okay. Hello, hello, can you hear me? Okay. So I'm going to be talking about some research that's at the intersection of algebraic geometry and mathematical biology. So let me just dive in to talk about what is phylogenetics about. So the main problem in phylogenetics is this. You're given a collection of species, and you would like to find the tree that best explains their evolutionary history. So like if I had 3 species, chimp, gorilla, and queer algebraic geometer, you would be trying to figure out, which of these 3 binary trees is the one that explains the relationships. Of course, this is kind of a toy example. Biologists know which of the 3 trees is the correct one. It's the middle one, where chimp is closest to human. Usually, we're interested in more complex phylogenetic trees than that. So here's an example of a kind of more realistic type of phylogenetic tree. And also, this is my kind of only GLBT vague reference. So this is a phylogenetic tree of gall wasps. And if you didn't know, Kinsey, the famous sexology researcher, before he did that, he was an entomologist, and this was the group of species that was his area of expertise, the gall wasp. But this gives you some idea of the type of tree we're looking to reconstruct. These phylogenetic analyses are useful both on short time scales and on long time scales. So that wasp phylogeny, that was on the order of tens of millions of years. But you can also do much shorter time frame phylogeny. So on the left is a phylogeny just within humans. So they extracted some DNA sequences from people living in sub-Saharan Africa, Eurasia, East Asia, the Americas, and Oceania, and constructed a phylogenetic tree. And that phylogenetic tree actually provides evidence for this out of Africa hypothesis. So like the sub-Saharan Africa is like it's separate from this sort of subclade that immigrated out of Africa. And you can go on even shorter time scales. So that the human phylogeny, that's on the order of tens of thousands of years. But on the right, we have a phylogeny of avian flu viruses. So this phylogeny is on the order of year. Okay, and so what the researchers were interested in when they constructed this phylogeny of different bird flu strains, they were trying to determine whether or not the different outbreaks of bird flu that happened in Nigeria were all caused by a single introduction or if there were multiple introductions. So you notice there's some different subgroups here that are pink, and those are all the strains that were extracted from these different infections, outbreaks in Nigeria. And you can see they're pretty distantly related to each other in the tree. And that provides some evidence, some forensic evidence that it wasn't just a single outbreak, but there were multiple introductions in Nigeria. Okay. How are phylogenies constructed? So the most traditional method, probably the method you're used to thinking about from your high school biology class, is that you compare basic characteristics of the organisms you're looking at. Maybe you make measurements of different features, like how big is the jaw, the shape of the skull, things like that. Look for the presence and absence of different features, and from that you try to reconstruct a phylogeny. That's something that, that goes back to Linnaeus, Darwin, etc., that approach. But we know that looking just at these morphological characteristics, that can lead you astray. So here's a kind of classic example. Of course, we have a big separation in the group of mammals, that we have the placental mammals and the marsupials. But if you were just to look at gross characteristics of size and basic features, you would make this comparison and think, oh, like the North American mole and the Australian mole are really closely related to each other because they fill the same ecological niche where they live. They're the same size and shape. They have a lot of the same features. They eat kind of the same thing. But in fact, there's this big divider, right? That there were the placental mammals that were separate from the marsupial mammals. So that kind of convergent evolution that we see when we just look at morphological characteristics can lead us astray, and that leads us to kind of more quantitative approaches to phylogenetics, which is what the majority of this talk is about. So the way we really build phylogenetic trees now is based on looking at sequence data from the different organisms you're looking at, and it's based on this basic principle that DNA sequences of related species are similar to each other, and more closely related species should have more closely similar DNA sequences. So if you want to build a phylogenetic tree, you extract DNA sequences from genes that appear in all the species you're looking at. So here's some kind of made up DNA sequences. Yeah, so my training's in algebraic geometry, so I do data analysis, but I like to talk about it. So here's some DNA sequences, pretty short, from human, chimp, and gorilla. And then the first step in the process, which is very non-trivial, but I won't talk about much, is to align the DNA sequences. So even though they may be code for very similar genes, those DNA sequences might not even be the same length. So you need to figure out which positions correspond to which other positions in the DNA sequences, and that's called the alignment problem. And so that's the first step, which I'll never talk about again. And after you've had the alignment, there's some probabilistic models of the mutations that happen to the DNA sequences that are then used to try to reconstruct the phylogenetic tree. So there's some models that I'll talk about in a second for how just single position changes happen in these sequences that will then be used. Okay, so what do those phylogenetic models look like? So they're examples of something in statistics we would call a latent class or hidden variable graphical model. The underlying graph is just the tree. And the way these work is you have a random variable at each node in the tree, and that random variable can just take one of 4 possible states. So it's either a C, a G, or a T, okay? So this is just modeling what's happening at a single position in the DNA sequences. And what you get for the whole long DNA sequence is just kind of you treat each of the sites independent from each other. And how does this model look just at a single site? So we have these random variables, X, and the random variables at the internal nodes of the tree, those correspond to species whose DNA we don't have access to. Okay, so usually the ancestral species, we don't have access to their DNA. DNA doesn't hang around very much. So like Jurassic Park is not real. Like you cannot extract the T-Rex DNA from a mosquito in amber. So we only have the DNA for the species that are around today. Those are corresponding to leaves. So in this model, these internal nodes, which correspond to theorized ancestral species, those are going to be hidden variables in the model that we don't get to observe. And the model is just a very simple Markov process model, which I'll explain how it works now. Whoops, So there was some ancestral sequence at the root of the tree, and there's some probability that at an individual site, it's in one of those 4 states, A, C, G, or T. Okay, so we have a probability distribution for what that state is at the root of the tree. Okay, and then along each edge, I just have a little Markov chain for how I change from one character to another along the edge of the tree. So there's some probability distribution for going from Y1 to Y2 along this edge. And then there's some other probability distribution for going from Y1 to X1 along this edge, and so on. And everything on edges is just happening independently of each other. So the probability that I would observe Y1, Y2, X1, X2, X3 at those 4 nodes in the tree would just be this product of probabilities here. But since I don't get to observe what state my sequences are in at those internal nodes, because they're hidden variables, you have to sum out over those states. Okay, and that gives me the probability of just observing X1, X2, X3 at the leaps. Okay, now you all thought you were coming to an algebraic geometry conference, and why is this guy talking about probability? Well, as an algebraic geometer, when I look at this, I see this is a polynomial. Actually, it doesn't maybe look like a polynomial right now, but look, I'm just multiplying 5 things together, and I'm summing out over Y1 can be in 4 states, Y2 can be in 14 states. So this is a polynomial of degree 5 with 16 terms. Okay, so I, as an algebraic person, would write that same expression in a more kind of polynomial way. So I have an indeterminate P sub I1, I2, I3. The I1, the I2, and the I3 are like, they can be one of 4 possible states. So I get 64 different probabilities. Each one of them is really a polynomial of degree 5 with 16 terms in these variables over here, which are the parameters of the model. Okay, so let me elaborate on that a little bit. So what our algebraic perspective is on these phylogenetic models. So once we fix a particular tree with n leaves and some underlying model structure, we're going to get a map that goes from some parameter space, capital theta, which is usually it's a polyhedron, more generally some sort of semi-algebraic set, and it maps into 4 to the n dimensional space. There's one coordinate for each possible n tuple of ACGT characters. Okay. The theta, again, is the parameter space of numerical parameters of the model, so what those different transition matrix probabilities can be. And those formulas for what the probabilities are, they're usually either polynomial functions or rational functions in terms of those parameters. So, okay, and again, this is just kind of what it looks like in the particular example tree I had on the previous slide. So then I can think about the model, which is the image of this map, as some subset of this high dimensional, 4 to the n dimensional space. So it's some, complicated semi-algebraic set sitting inside of R4 to the n. And we want to study that object. Okay, and so, we'll use algebraic geometric tools to make this study. So we're going to have the polynomial ring in those 4 to the n different elementary probabilities, those 4 to the n different indeterminants, P sub I1 up to IN for each n tuple of ACGT characters. And then I have the image of this map. So that's sitting in 4 to the n dimensional space. So I can, compute the vanishing ideal of that image. And that object, that ideal, is known as the ideal of phylogenetic invariants of the tree. Why are they called invariants? Biologists came up with that name. Of course, they didn't know anything about ideals and stuff like that, but they introduced this concept of like having polynomials that evaluate to 0 on a phylogenetic model. So we have this concept of phylogenetic invariance. And of course, the model is a semi-algebraic set, but of course you can think about just what's the variety of that ideal, and that's we would call the phylogenetic variety associated to the tree T. Of course, the model is a subset of the variety. For most interesting models or models that have a close enough to an algebraic structure to them, one thing that's really nice is that closure of going to the Zariski closure doesn't increase the dimension. You end up with something that has the same dimension that's kind of useful in some proofs and things. Okay, so let me just try to give you one example of what these equations look like and how we use some kind of basic ideas to figure out what some of the equations look like. So here's a tree with 4 leaves, and the probability of observing LMNO at the leaves across the top here, well, it's going to be a polynomial of degree 7, and it has 64 terms, and it's this polynomial here. Okay, and I want to find some polynomials in the p's that evaluate to 0 for any choice of the a's, pi's, all of this stuff here. Okay, so when I plug in those, I want it to evaluate to 0. And the idea behind how we're going to find some of these equations, some of these phylogenetic invariants, it goes back to some computational biology thought process about how you actually compute the value P, L, M, N, O. You don't want to actually expand this polynomial into 64 terms and then sum up all those terms. In general, if you had n leaves, this polynomial would have 4 to the n terms, more or less. So it's like, it's way too big to expand and compute it that way. But you don't have to do the sum as it's written here. You can rearrange things. So in phylogenetics, they call that the Felsenstein's pruning algorithm. In algebra, we would call that the application of the distributive law. But how does it go? So I color-coded the indices. I don't know how well you can see it in the audience, but I color-coded the indices like as they appear. So the I, the J, and the K, which correspond to these internal states here. So the J and the K, so the J's corresponds to this one and the K's corresponds to this one, they never appear in the same variable. So they're kind of completely independent from each other. So I can group all the J things together. So that's this term here. And then separately, I have all of the indices that involve K. So now I'm multiplying all of those together. So now this expression, once I summed out by J, is like a 4 by 4 by 4 tensor. It's indexed by I, L, and M. This expression over here is another 4 by 4 by 4 tensor that's indexed by I, N, and O. And now I'm going to multiply them together by their I indices and sum out, like so. And now what we do is we're going to think about, instead of thinking about the result as a 4 by 4 by 4, 4 by 4 by 4 by 4 tensor, I'm going to think about flattening it down to a matrix. Okay, so I have the LM indices, which only appear here, and the NO indices only appear here. So if I group the L and the M indices together, so that for each value of I have a vector of length 16, this expression, oh, and over here I have a vector of length 16, a row vector. So this expression is really telling me, take the outer product of those two vectors together, and then, of course, that's going to be a matrix of rank 1. I add 4 of those together with these weights pi, and so that's going to give me a matrix of rank less than or equal to 4. Okay, so just some basic linear algebra plus the distributive law gives us some conditions on these matrices that have to be satisfied for any probability distribution that comes from this model. And of course, that is the rank condition gives me some equations in the vanishing ideal, because if the matrix has rank less than or equal to 4, then all the 5 by 5 subdeterminants of this matrix should belong to the vanishing ideal. Okay, you can generalize this to arbitrary trees. It's not something that only holds on trees with 4 leaves, and you just need a little bit of combinatorics about trees. So we have this notion of a split of the set of leaves, which is just a bipartition of the set of leaves into two disjoint sets. And, a split is called valid for the tree T if it comes from removing an edge in the tree. So if I in this tree here, if I rip out either one of these edges, it separates leaves 1 and 2 from leaves 3 and 4. So that gives me a valid split 1, 2 from 3, 4 in this tree. But this split 1, 3 from 2, 4 is not valid in the tree because like the subtree indexed by 1 and 3 and the subtree indexed by 2 and 4, they intersect each other. Okay? And the general theorem you can prove is that for each one of these splits, there's also an associated flattening of this tensor, which is giving you the probability distribution. And in general, if you have a valid tensor for your tree T, any probability distribution that comes from the tree T, the rank of that associated flattening matrix will be less than or equal to 4. And conversely, if the split is not valid for the tree T, then generically the rank of that flattening is bigger than 4. So these rank conditions, or 5 by 5 minors, they distinguish whether or not the tree has that split or not in it. Okay, so that's kind of like a high-level overview of kind of what is phylogenetic algebraic geometry about, and some of the examples of some of the equations that belong to the vanishing ideal. If it was too much, just think, all I really want you to know at this point is to each tree, there is a variety. And there are equations that evaluate to 0 on that variety. They are called phylogenetic invariants. That's really all that you need to know to understand what's going to come after this. The point of studying this field, phylogenetic algebraic geometry, is that trying to make sense of what those varieties are like and what the equations in the vanishing ideal are like is useful and interesting, okay? It's useful because you can use it to build new algorithms to reconstruct phylogenetic trees from data, and I'll try to give you a little taste of that. It's also useful as a theoretical tool in phylogenetics because it allows us to prove some new identifiability results, which I'll talk about what that means. And maybe more importantly for this conference, one of the reasons they're interesting is there's lots of cool math that comes up in the study of these varieties. And I'll try to emphasize some of that towards the end. Okay, so let's talk about using the phylogenetic invariants to reconstruct trees. So the basic idea and the reason that this whole field exists is because some biologists had this idea of saying, oh, there are these equations that evaluate to 0 for each tree. If they satisfy this extra feature that they're phylogenetically informative, which means the invariants for one tree, they evaluate to 0 on that tree, but they don't evaluate to 0 for distributions that come from a different tree. You can use that as a tool to figure out which tree is the correct tree for your given data set. Those invariants are called phylogenetically informative if they have this property that they distinguish between a tree and some other tree. And so the idea of Cavender and Felsenstein and Lake, so biologists, was, well, you have these invariants, you're just going to take some empirical estimate of what the true underlying probability distribution is just by taking your aligned DNA sequences and counting the number of times each of the different columns arises in that alignment. You just plug those into the phylogenetic invariants. If the distribution exactly came from the model, it would evaluate to 0, but since you just have an estimate of the distribution, it should evaluate to something close to 0 if it came from a tree and something far from 0 if it didn't come from the tree. So that's their idea. There was a lot of kind of back and forth in the kind of phylogenetics community about whether or not that was a good idea and how to do it. I'll just say that if you think about things carefully, it's possible to construct methods based on this idea that actually perform very well in practice. But they tend to work just for small trees, just because, well, you'll see why in a second, maybe. Then, once you've reconstructed trees just with 4 leaves, you're going to use some combinatorial tool, and in phylogenetics, that's called phylogenetic puzzling, to put together. Let's say you have like 10 species you're looking at. So you look at what trees you can reconstruct on just all the 4-element subsets of the species. You reconstruct a tree there, and then you try to put them all together into a bigger tree. And so there's a bunch of different authors who have worked on that side from the algebraic perspective. And here's some of the authors that are involved in that. Here's an example of what that looks like, and this is probably like the most famous and successful method for doing this. It's called SVDQuartets of Chitman, Kubatko, and Long. And it's for a more complicated model than the one that I've told you about so far that involves this extra thing called the multi-species coalescent. Still, the phylogenetic invariants that they end up using correspond to rank conditions from the flattenings, but it ends up being not rank 4, but rank 10 is the special rank of interest. And so what are you going to do to decide on 4 leaves which of the 3 possible unrooted trees is the best tree that explains your data? You want, for each of those possible splits, you want to test, is my given matrix close to a matrix of rank 10 or not? And you don't do that by evaluating all the 11 by 11 minors of this 16 by 16 matrix. That is a really horrible way to test if a matrix is close to being a rank 10 matrix. What you do is you use some tools from numerical linear algebra. In particular, you can compute the singular value decomposition, that's why it's called SVD, to determine how close you are to being a rank 10 matrix, and then you pick the tree that best looks like it's a rank 10 matrix from the singular value decomposition. Okay, so that's the basic idea of how you use these algebraic tools to develop new methods for reconstructing phylogenetic trees. In the end, you don't actually use the algebraic geometry in the sense of you're not like plugging into polynomials, but you use the ideas that come from algebraic geometry in terms of figuring out what these rank constraints are and the different phylogenetic invariants are to lead you to some new algorithm. Okay, the second thing that this perspective on phylogenetic algebraic geometry is really useful for is for proving results about identifiability of phylogenetic models. And identifiability, all that means is that you have some statistical model or other type of model. Those models have parameters in them. And the model, you input the parameters, and then it gives you out some distribution. And what you want to know for the model to be identifiable is that you should be able to recover the parameters from the thing that you can observe, which is the distribution or an estimate of the distribution. So that's what identifiability is. So it's sort of the theoretical step you need to know is true about your model before you can then start trying to build a method to reconstruct the tree. If your model doesn't satisfy identifiability, there's no way that you can reconstruct a tree from data because multiple parameter choices give you the same tree, say. Of course, people in phylogenetics don't usually do that. They just come up with a model and they say, here's a method, it works to reconstruct a tree, but then they a lot of times don't prove identifiability. They just assume that's identifiable, and sometimes there are situations where it ended up that it wasn't identifiable, and that's a problem. Okay, so in phylogenetic models, there's 2 types of parameters that are of interest. There's those numerical parameters, which are the transition probabilities in the model, but then there's also a discrete parameter, which is probably the more important parameter, which is which tree produced the probability distributions, right? I said you fix a tree, and then you have all the numerical parameters that give you all these probability distributions. But you really want to know which tree is the right tree. So that's a kind of parameter. And I'm mostly going to talk about the tree parameter in terms of this application now. Okay, so what's our geometric perspective on what identifiability is about? So you have this tree parameter in a phylogenetic model, and you get a family of probability distributions. And for the model to be identifiable, what you want to know is that for all probability distributions that belong to the model, there's not some other tree such that the probability distribution belongs to the model for that other tree. So once you have the distribution, you can tell which tree it came from. So geometrically, the picture we want to have is this situation that for each different tree, you're getting a different disjoint set of probability distributions. You definitely don't want to see this bad situation over here that like one is nested inside of the other or something like that. Okay, now it's known in phylogenetics, probably should be better known, but most of the math people know this, that identifiability is too much to ask for in most of these models. And that's because they actually can intersect each other. That's what we usually see in these models. And so we weaken this notion to generic identifiability, which there's different ways to phrase this. This is the way that I kind of prefer to do it, which is the condition that you want that for any two different trees, you intersect the models for those trees. And what you'd like is that the dimension of that intersection should be strictly less than the dimension of either of those two models. Okay? And so what that means, again, geometrically, is that you have intersections between the models for the different trees, but if you were to randomly choose a point that was on the union of the different models, then with probability 1, it's not in the intersection. That's what this condition means. So with probability 1, we should be able to tell which tree produced the given probability distribution. Okay, so how do we use algebraic geometry to figure out whether or not the tree parameters of a model are identifiable? It's just based on those invariants that were the phylogenetically informative invariants. Remember, phylogenetically informative invariants, those are ones that evaluate to 0 for one tree, and they don't evaluate to 0 at every probability distribution in another tree. Okay? So if you have phylogenetic invariants that are phylogenetically informative, so that is, you have an invariant that evaluates to 0 for all probabilities in the one model, and it doesn't evaluate to 0 for some distribution in the other model, because these semi-algebraic sets are images of polyhedra under polynomial maps. Like the Zariski closure is an irreducible variety, so that automatically implies that the dimension of the intersection will be strictly less than the dimension of either. Okay. So if you can just find phylogenetically informative invariants, then you know that the tree parameters of the model are identifiable. Okay, great. Well, so we can immediately use this to prove identifiability of these phylogenetic models that we mentioned. Because, okay, so theorem, the unrooted tree parameter of phylogenetic models is generically identifiable. How does this work? Remember, we had these rank conditions that come from edges in the tree. So you take an edge in the tree that gives you a valid split. You look at the flattening of the matrix. The rank of that should be less than or equal to 4. If the split is in the tree and it's greater than 4, if the split is not in the tree. So those edge flattening invariants will tell you exactly which splits are in your tree. And then there's a combinatorial theorem that you prove that says once you know which splits are in your tree, that uniquely determines the tree. Okay, so this theorem, so So I didn't put anyone's name on it because, I mean, people know this result for the basic models that that I've told you about so far for a long time, using more kind of traditional methods in phylogenetics, not using algebraic geometry. So this theorem in and of itself is not that interesting. What's interesting is that you can apply this same idea to much more complicated models where people could not use the standard approaches in phylogenetics to prove identifiability. So I just have some examples of different types of models, which I won't go into detail about how those models work, but they're models where either the evolutionary process is much more complicated, so you don't just have 4 states, but you have more states of your random variables for different reasons, or they're models that involve having not just one tree, but multiple different tree structures, taking into account different types of evolutionary processes like hybridization and gene transfer and things like that. The same approach can also be used in a completely non-phylogenetic context, but to problems of identifiability come up all over applied mathematics, so also in the study of ODE models, which I've done with various co-authors. And one kind of more recent thing with one of my students, Ben Hollering, was to kind of realize that this idea is really, well, you can think about it as a statement about the matroid associated to the variety. There's an algebraic matroid associated to each variety. And so you don't even need to find equations if you can just understand the matroids well enough to prove identifiability. So there's kind of lots of kind of interesting questions here, and there's lots of further problems to apply it to. Okay, so for the rest of the talk, what I want to talk about is more about the kind of connections to algebraic geometry, so sort of more deeper connections. Actually, maybe before we do that, I feel like I'm going a little faster than I was expecting. Are there any questions at this point before I dive in more into the algebraic geometry?
For the models we were talking about on the other page, do any of them have diagrams at all resembling phylogenetic models, or is the connection less visible?
So, I mean, it depends on which model you're talking about. So the covariant model, there's still a single tree. It's just you have a more complicated structure of the way the random variables can be related to each other in the tree. Mixture models, those are ones where you have just multiple trees. So there's not just a single tree, but you have a whole collection of different trees and things interacting that way. Profile mixture model, usually there's just a single tree, but again, it's a more kind of complicated interaction about how things are related to each other. The ODE models, yeah, there's usually often like a graph or something like that. It's not a tree, but there's some kind of graphical structure like that. And there's one big active area in mathematical phylogenetics right now is to try to not have trees at all, but to have more complicated network structures to take into account those like hybridization events. But usually, yeah, there's a graphical combinatorial representation of the model. Yeah. Yeah. Thanks.
So these often have is it specific to these categories, or could one ask for other methods? I mean, could one give an approach for other methods? For example, in our neural networks, they have big very big all these edges in neural networks, and they are also horrible with other methods. Could someone switch to similarly, or?
Yeah, absolutely. So, I mean, the techniques, so like I was saying earlier, like the phylogenetic models, at least the most basic ones, are examples of what are called graphical models. That class includes lots of things like neural networks and other things like that. So absolutely, questions about identifiability are relevant for all types of statistical models that people use, and these techniques are relevant to those types of models as well.
Are there nice results?
I have not worked on that. There are people in this algebraic statistics community who have worked on things like that. So yeah. I mean, the basic statement that the intersection of two things is, if you can find an equation, is lower is just a statement of algebraic geometry, which can be applied to all kinds of contexts. It's a question then of, okay, you have your specific model you want to look at, then you need to analyze the algebraic geometry of it and find what the equations are. So. I'm sorry, I couldn't hear that.
So in this case, I would have identified mistake and zero and one have to be so that final is better enough, but I would use the final.
Yeah, so I mean, again, the condition is it evaluates a 0 on one and not the other, so it's a pretty, yeah. Okay, let me kind of dive in now to the more of the algebraic geometry side here. So the basic question just on the algebraic geometry side is to, again, characterize what are these equations and try to understand what these varieties actually look like. And there's this, this structure theorem that was sort of, so Baron Sternfels and I had proven this in a special case, and it was sort of successively generalized until eventually Drysma and Cutler had the kind of most general statement, which, again, leaving things somewhat vague, says if you want to know what the vanishing ideal is for an arbitrary tree, you just need to understand what the vanishing ideal looks like for three-leaf trees in the case of binary trees, or more generally trees that have just a single internal node and k leaves, if you want to understand it for arbitrary tree structures. The varieties that come up for those star trees are often kind of interesting classical algebraic varieties. Sometimes they're toric varieties, sometimes they're secant varieties of toric varieties. There's this one example that comes up that some reason got a ton of attention, which was the fourth secant variety of the Saugré product of 3p3s. But anyway, so there are varieties like that, classical kind of combinatorial algebraic varieties. So how does this this general theorem of Drysma and Cutler work? The basic idea is there's a kind of structural result that says if you want to understand what the vanishing ideal for this tree is, which is obtained by kind of gluing the two trees together at a leaf and then like suppressing that internal vertex that you glued at, there's a big group action that comes into play, which is that remember there's like a matrix associated to each edge of the tree, right? That was this Markov transition matrix. So at the leaves, you just kind of have that matrix hanging there off the edge, and you can like multiply by an arbitrary matrix, and that won't change the variety you get, right? So there is this GL action, or more generally, it's actually like some more complicated group usually than just general linear group, but it acts at each leaf. And the thing that you, of course, it's acting on the variety, but it's acting on the coordinate ring as well, since that's kind of what we want to understand. We want to understand what these equations are. And then the thing that you observe is that actually the coordinate ring for the big tree is actually just the invariant ring of that action on the two components, where you have it acting by G on one side and G inverse on the other. So in other words, the variety for the big tree is just a GIT quotient of the product of those two varieties, quotiented out by this algebraic group. And so what that means in terms of understanding what the equations on the variety are is that You can take the vanishing ideal for the two smaller trees. There's a way you use that group action to extend those equations to the bigger tree, and then you have to throw in a bunch of these minors from flattenings that come from that edge that you glued upon. Anyways, the proof, it uses just some classical invariant theory, so going back to the early 20th century. And so one of the things that I've done with some of my collaborators is to try to extend that idea to other contexts, not necessarily related to phylogenetics, but there's lots of situations where you have some variety they're parameterized by graphs in some way, and then you can do like graph operations where you join things together, and there's maybe some kind of group action, and you can think about how does that relate the ideals of the components to the ideal corresponding to the bigger graph. And so depending on the context, you might get a perfect analog of the previous theorem where you actually get that the coordinate ring of the bigger graph is isomorphic to the invariant ring of the tensor product. But usually it's not isomorphic, but you can say something like it's the subring of that tensor product that's generated by the degree 1 pieces. And then by analyzing combinatorics, sometimes you can say stuff to figure out what the vanishing ideal of the big graph is. So the case that we understand the best is the case where everything is like toric. So that is this group here is really just an algebraic torus acting. And there you can have a lot of combinatorics where you can kind of get access to what those equations are. So again, there are lots of different classes of graphical models that are interesting to study in applications, not just algebraic statistics. But if we want to get our hands on the equations, having tools like this can be really useful to understand those things better. Let me talk a little bit more about like what those equations look like in a particular class of phylogenetic model. These are models that are called group-based models in the kind of mathematical phylogenetics community. So one of the things that I said a few times, but probably I never defined what it meant, was once you specify a model structure, then you get a variety, blah, blah, blah. But I didn't ever tell you what that meant. So the model structure is that you can place some restrictions on what the transition matrices look like in your model. So the model I was mostly talking about earlier was a model where you just have no restrictions at all. They're just arbitrary Markov transition matrices. That's a model called the general Markov model. But in these group-based models, we have very restricted transition matrices that and there's essentially 4 of these models that people actually use in phylogenetics. There's a model not for DNA sequences, but for just binary characters, where you just have a symmetric transition matrix. It looks like this. So there's beta, which is the probability of swapping from a 0 to a 1, and then alpha, the probability of staying at whatever state you're at. And then these other models, the Buczynska-Wisniewski model. This one's called the Komura 2-parameter model. This is the Komura 3-parameter model, and on that one, maybe you see why they're called group-based models because, hopefully this looks familiar from your undergraduate abstract algebra class. It's like the multiplication table of the Klein 4 group. And what the structure really is, besides just seeing that you have like the multiplication table of some finite group there, is that if instead of the random variables just being ACGT, unstructured, you replace those with the elements of this finite group, in this case, the Z2 cross Z2, then the structure that gives you this shape here is that the probability of transitioning from one group element to another is just a function of the difference of those two group elements in the abelian group that you're looking at. And so what that means in practice then is that the probability of observing these particular this n tuple of group elements at the leaves is just, again, it's that sum over all the internal states. But the functions that are involved are all functions of differences of group elements. So, if you ever took a class that had Fourier transform in it or something like that, then this is screaming that you have a convolution of a bunch of different functions and that the first thing you should do is take the Fourier transform to simplify things. There's something that was noticed by some statisticians who were studying this problem, and then when Bernd Sturmfels and I were looking at this, then we translated that to say, well, after you take the convolution, you're just getting after you take the Fourier transform of a convolution, you just get a product of the Fourier transform. So that means that the model can be parameterized in this Fourier coordinates as a monomial. So you're just looking at toric varieties in this case. So these group-based models are all just toric varieties, but not in the natural coordinate system, which is the probability coordinates, but in these transformed coordinates, which is like the phase space or whatever. So again, for the very specific model of this model that just has two states. So this model is called the Cavendish-Ferris-Neyman model, this binary symmetric model. The structure is especially simple there. And the equations just turn out to be, they're always determinantal equations, and they're always just 2x2 minors of a bunch of matrices, okay? So you have a tree. Here's what the Fourier transform looks like. Maybe you're scared of Fourier analysis or something, but in the discrete case, it's just a linear change of coordinates, okay? All the coefficients are 1s and -1s, okay? So the new coordinates are called the Q's, or I don't know why, but that's what we call them. Or that's the phase space coordinates. And then for this four-leaf tree, for this Cavendish-Ferris-Neyman model, it turns out that the generating set is just all 2x2 minors of these four matrices. Okay, so they have a very, very simple structure in those coordinate system. Yes, the queer coordinates. Okay, so you can ask deeper questions then about these varieties. They have such a nice structure, the equations. So maybe like more interesting stuff about the varieties is true too. And so of course, I think everyone here knows what the Hilbert function is and the Hilbert polynomial. So you take the coordinate ring and you look at the dimensions of the different graded pieces of the coordinate ring, and that gives you the Hilbert function, which eventually is a polynomial called the Hilbert polynomial. And Buczynska and Wisniewski, they knew that these things were toric varieties, so they're like, oh, well, let's analyze more deeply the algebraic geometric structure. And the thing they realized is that for this, again, very simple model, the Cavendish-Ferris-Neyman model, for any trivalent tree with n leaves, all of those Hilbert functions are the same, regardless of what tree that you start with. And the varieties are not isomorphic to each other, unless the trees are actually, isomorphic to each other under some permutation of the label. So there's something kind of non-trivial going on about like why are these varieties related to each other. So again, what we're saying here is that in the Hilbert scheme, which parameterizes all quasi-projective schemes that have the same given fixed Hilbert polynomial, each tree is giving you a different point, and they're all in the same component of the Hilbert scheme. And the way that Buczynska and Wisniewski proved this theorem was by doing these kind of like flops or something. That's probably, I shouldn't have said that because I know that means something in algebraic geometry, but they do this like local change to the tree that changes it from one tree into another, that then they get a whole family of things that don't correspond to trees that all have the same Hilbert polynomial. And then they're able to show that they can get from any tree to any other tree by using those kind of deformation degeneration sequences, okay? So of course, the thing you would hope, though, is that maybe there's just one thing that degenerates to all of the varieties corresponding to trees. Like we know examples like that, right? Like the Grassmannian of planes, that degenerates to toric varieties, one for each tree. Like that's what the whole like tropical Grassmannian thing, at least for G2n, is about, right? So this seems like it should be somehow related to that. And what Sturmfels and Zhu had proved is that that is true, but it's not for the Grassmannian. In this case, it's for this variety, which is the, well, let's say on the ring side, you take the Cox-Nagata ring of the blowup of Pn-3 at endpoints, and it's that ring that degenerates to all of these coordinate rings for each of the different trees. And the proof sketch uses Kastrovet and Tevelev's results on what that Cox ring looks like, connections to the kind of Hilbert's 14th problem. Also, the Verlinde formula from mathematical physics comes up into the argument as well. And then lastly, there's this notion of Khovansky degeneration or Sogby bases that is useful for putting all these things together. And this has been generalized somewhat by Chris Mannin to beyond trees, but still in this kind of CFN model situation. But there's lots of other situations where you could ask, does something like this work? We have all kinds of different families of models. Each one has a tree. Like, what do the Hilbert functions look like? Like, what's the analog of this, of the Grassmannian or this, Cox ring or whatever? How do those things all fit together? I have no idea if it has any application to phylogenetics, but it's really just kind of interesting algebraic geometry that is related here. Okay, so let me just summarize what I tried to tell you in this talk. So phylogenetic models, which are kind of statistical models used in mathematical biology, they really have this kind of fundamental algebraic geometric structure to them because they are parameterized by polynomial maps. That algebraic perspective is useful for developing new algorithms for reconstructing phylogenetic trees from data. It's also really useful for proving theorems about identifiability. And I didn't really spend a lot of time in that section, but our best results for some classes of models really come from this algebraic perspective. And it also leads to lots of interesting new mathematics, like I told you about at the end. And there's lots more to do in this area. Anyone who wants to get involved in projects is always welcome to join this community. And that's all I wanted to say. Thank you so much for your attention.